our fourth televisitor to my freshman writing class, the scholarly, what was it called again? SSI? Seminar and Scholarly Activity. Thank you. Inquiry. Thank you. 165, Symbiosis and Parasitism in and around us. My goal in this course is to teach some basic skills in writing and analysis of the literature, and it was my decision to use symbiosis and parasitism as a fulcrum to teach these kinds of concepts. And I've been extremely fortunate in having a number of fabulous scientists decide to televisit with us as people begin to eat the food I brought in, which is great, right? And so what I would like to do now is invite an electronic pal of mine, Dr. Irene Newton of Indiana University, who has what is the finest and most fabulous name for a lab website I can think of, Disco Genome. What can I say? And has worked in a number of different areas of interest to me, though apparently she studies in part the great pandemic, as Ed Jan puts it, of Wolbachia. She is also well experienced in deep sea symbioses, and though it is a bit smelly, some of those sulfide-based ones in particular. And it's on this subject of deep sea symbiosis that Dr. Newton comes and chats with us today. And I would like to introduce Dr. Newton to my class, and then she can address the questions you all were kind enough to send her based on the readings. Dr. Newton, my class.
Hello again. So I thought that maybe I would start by telling you a little bit about the background necessary to understand some of this metabolism, because some of your questions were just asking, give us a little bit of a primer on what's going on here metabolically, and that then we could get started on the questions. And feel free at any time while I am giving you this primer to just stop me, say I can't see what you drew, or ask any questions about what I'm going to talk about. So one thing that I think we as scientists, and just as human beings on this planet, are guilty of doing is seeing the world through our human glasses, right? So we have human vision, yeah? I would say you're all human, and the things that you see around you are colored by the fact that you are human. So when people say life at extremes, what they really mean is life where humans can't live, right? And when people say, that seems like a difficult, it seems difficult to think of life existing on other planets, they think about that in the context of the kinds of requirements that are needed for human life, for the kind of things that we need. So I wanted to use that as a preface to begin to talk about what is necessary for life, at least on this planet, what we know organisms use for life. And so have you guys seen this kind of little chart before?
Oh,
No.
I like that.
All right, so every single organism that we know of on Earth requires a source of energy, a source of electrons, and a source of carbon for life. Now, you guys, let's start with plants, because I think that's, most people can get the answers for plants pretty easily, because I think photosynthesis is drummed into your heads at an early age. Where do plants get their energy from?
Sunshine.
Very good, very good. And what about their carbon? Sorry?
Sing out, folks.
Carbon dioxide.
Carbon dioxide, that's right, because they fix their own carbon, so they get it from carbon dioxide, and then they need electrons in order to turn that carbon into sugars. Do you know where they get the electrons from?
Water.
Water, excellent, yeah, so good for you. So plants get their energy from light, their electrons from water, their carbon from carbon dioxide, so we call them photo, litho, autotrophs. And I'll write that out for you on this chart here. Yeah. So photo meaning energy from light, litho meaning an inorganic source of your electrons, autotrophy meaning that you make your own sugars by fixing that carbon. Now, what about you? Have you thought about this for yourself? So where do you get your energy from? Sorry? Food, yay! And what about your electrons? Food! Okay, and then what about your carbon? Yeah, you're very boring, aren't you? It's food all the way across, right? So you are the opposite of plants. You are chemo, organo, heterotrophs. So I'll write that out as well.
By the way, I love this. I haven't seen it done this way. Thanks.
Oh, yeah, well, I have to credit my advisor, Colleen Kavanaugh, for this, because this is how she teaches chemosynthesis. So chemo, organo, heterotrophs, because you get your energy from chemicals that you eat, you get your electrons from the organic chemicals that you consume, and you're a heterotroph because you consume the carbon of others. You don't make your own carbon, right? So what you see here in terms of plants and you, that is the metabolic diversity that we find in the majority of metazoa on the planets. So metazoa meaning multicellular life on the planet. So most of the multicellular animals and plants that we observe around us are chemoorganoheterotrophs or photolitho autotrophs. But as you may know, bacteria can actually be any combination of these things, and they can be facultative and switch back and forth. So you can find chemolitho autotrophs like those at the deep sea. You can find photoorganoheterotrophs or photoorganoautotrophs, just any combination of these different metabolisms. So they can use different substrates that are around in the environment as an energy source or as an electron source and as carbon sources. So they're extremely versatile, which is why they have colonized so much of the planet, and why I would argue that they potentially have colonized outside of Earth, other planets as well. So is that pretty clear? Little chart, we can move on. The second thing that someone asked me about was just explaining how chemosynthesis works in general. And for that, I'm going to put up this other little chart, which is comparing the photosynthetic process in a plant, or here a tree, to that in Riftia pachyptila, the tube worm. So while for the plants, they're getting their electrons from water, they're getting their carbon dioxide from the air, and the energy is from the sun. Do you guys remember how they actually harvest energy from the sun? Anyone? Chlorophyll, that's right. They're photosystems. And in Riftia's case, what we have is carbon dioxide being taken up from the water. We have both oxygen and sulfide being taken up from the environment, and they're being produced by these volcanoes under the ocean. So it's actually the geothermal energy coming from these the rifts that are actually producing the chemicals necessary for these microorganisms and for the symbiosis to exist. And the bacteria actually, oops, there go my Western blots. They use the energy of the oxidation of sulfide with oxygen and the electrons derived from that to be able to fix carbon dioxide. So they are in many ways similar to plants, to the photosynthetic process, but they're using a completely different source of energy and electrons. Okay. Any questions about that? No. All right. So I very much enjoyed seeing your questions, and I'll just kind of start at the top and see what I can get to from everybody's set of questions. So number one was Lauren. Who's Lauren?
Wave, Lauren.
Hi, Lauren. So Lauren asked an interesting question. So why do you think there aren't more multicellular organisms that rely on the use of hydrogenosomes instead of mitochondria? And so do you guys, from reading that article, do you know why or what is hypothesized to be the selective pressure for a hydrogenosome versus a mitochondrion? No? So all of these eukaryotes that people have found hydrogenosomes within, they are living in anoxic environments. So they're living in environments where we have very little oxygen. And it turns out, I mean, you might know from other classes, this is actually a difficult way of life. So living without oxygen, especially as a eukaryote, is difficult. And the multicellular organisms that rely on hydrogenates are even more rare. So it's not that difficult to believe that it might be something that is energetically difficult to maintain as a penicillin. So that would be my answer to that. And then I'll move on to Theo. So Theo asked, do you think the first life forms on Earth evolved in hydrothermal vents? And do you think that there are any connections between theories regarding primordial slime and certain environmental conditions in the ocean and those regarding underwater unicellular organisms living near hydrothermal vents? So a lot of people who study the deep sea think of it as a potential analog for the primordial Earth, or a place where life could have evolved in the primordial Earth. And the logic is that, as you can see from my little diagrams, that you have a source of energy, of electrons, and of carbon even without there being sunlight. And it's possible that life evolved on the bottom of the ocean. That said, I think to this date, there is not any evolutionary evidence that the organisms that live there now are so ancient as to have been there, first, like the first on the scene with regards to the evolution of life on the planet. And perhaps that is because the majority of the symbioses that are there now, and even the extracellular microbes that are chemosynthetic, they actually use oxygen to oxidize these sulfides and derivatives from the vent. And do you guys know when oxygen came on the scene on planet Earth? You guys know this? No? So for many, many years after the formation of the planet, the Earth was anoxic. There was not oxygen. But then when you had the evolution of photosynthetic prokaryotes, can I use that word, Mark, or are you an anti-prokaryote person?
Abigail Salyers and I invented the term prokaryotic pride. So Norm Pace and John Eisen hit me, but can we say it here?
I love prokaryote. I think it's useful.
Can I send you a prokaryotic pride button, though? Can you wear it?
I'll wear it. I'll wear it proudly.
Do you want the one that says free the organelles, or the one that says first evolved, last extinct?
Oh, first evolved, last extinct is good. Yeah. So going back to what I was saying, these photosynthetic prokaryotes that evolved on the planet then produced oxygen as a byproduct of photosynthesis. So because they were stripping water of electrons, they produced oxygen. And that actually oxygenated the entire planet. And that allowed organisms like us that have a mitochondria that takes advantage of oxygen, and organisms like those at the bottom of the ocean, the chemosynthetic organisms that take advantage of oxygen, to exist. Maybe that's a roundabout way of saying that it's possible that the conditions on the bottom of the ocean were such that there were life forms that evolved there first, but that the dominant communities we see there now are not likely to have been the relatives of those organisms, if that makes sense. All right. And I didn't get to wave at Theo. Where's Theo? Oh, hi, Theo. All right, Genevieve is next. Hi, Genevieve. So I really liked your questions because, again, it made me think about this idea that we see the world through our human-centric vision. So how do we know what is a habitable zone for extraterrestrial life, right? If it's not the same condition as on Earth, how do we know what to expect when we look at another planet? And we don't. We're just basing, the only example of life that we have in the universe is what we see in front of us, right? So we're basing all of our prerequisites based on what we know is necessary on Earth. It's an interesting question to ask also because, when people are designing these probes to go out into space and try to get samples back, how would you know if there was life there? Like, what's one way of looking? Well, they look for the macromolecular signatures of life that we know about, right? DNA, RNA, proteins, these kinds of molecules that we've identified on Earth, but potentially they could be completely different macromolecules on another planet, and it would be more difficult for us to detect given the mechanisms and the protocols that we use to be able to identify them. Okay, Gloria. Where's Gloria? Hi, Gloria. So I thought this was also an interesting question. Do you think chemosynthetic symbionts have descended from pathogenic ancestors? And I think many of us who work in intracellular microbes think about where these organisms came from and how they were able to invade the cell if they weren't initially mutualistic. It's possible that they did initially colonize and not have much of a mutualistic effect on the host, but then evolved one. That said, I should mention that the many endosymbionts, and where symbiosis is defined as pathogenesis on one end and mutualism on the other end, they use the same molecular toolkits to invade the eukaryotic cell. So they all have secretion systems that they can use to get in, and they likely secrete effectors and proteins to modify the cell as they're infecting. Just the outcome of the infection is different. So it's definitely cool to think of what happened initially for the symbiosis to occur. And especially in the case of Riftia, which is totally puzzling, right? You have an organism with no mouth, no anus, completely colonized by these microbes. And it's kind of a chicken and egg question. What came first? Riftia can't exist without this microbial community, or these microbes that colonize it inside of it. And how would an organism have developed this strange symbiosis? People who have studied the tube worm and have looked at really early larval stages of the tube worm have shown that it actually has a complete digestive tract during development. But then the bacteria colonize the tube worm. They actually come in from the outside, which is so bizarre. You might think, oh, the larvae are going through the ocean. This is a little tube worm larvae. It's trying to find its symbiont out there in the ocean, and the symbionts are coming in the gut, and then they're colonizing it from the inside. But no, instead, the symbionts actually adhere to the outside and then invade the tissue, causing this massive disruption in the tissue. And then the tube worm settles and becomes the adult tube worm with this massive organ called the trophosome that's colonized by those chemosynthetic organisms. So it's a very, in the way that it colonizes, it's very pathogenic seeming, right? Because it's coming in from the outside, it's causing all of this lysis of the host tissue. But we don't know. We don't know if it came from pathogenic ancestors or not. And but it's an interesting question to think about. I feel like you guys are not. If you have any questions as I go through, just kind of wave your hands around and I'll stop. Mary's questions are next. Where's Mary?
Oh, here comes Mary.
Oh,
There
hello,
she
Mary.
is.
All right. So Mary had a little Wolbachia slipped in here. She was like, I don't care if we're talking about the deep sea. I'm going to talk about Wolbachia because Wolbachia is the awesomest. I'm with you there, Mary. So is there any ecological or general danger in how widespread Wolbachia is in insect populations, especially in the intentional use of it to wipe out disease-vectored insect populations? This is something the Wolbachia community is worried about. People who study Wolbachia are concerned that this very pathogenic form of Wolbachia might be released, and because Wolbachia are known to be horizontally transmitted, that it could get into another insect that people care about, say the monarch butterfly, for example, insects that are not disease vectors. It is, also concerning this other, I should mention, the reason that people are using Wolbachia is to both decrease the population of insect vectors and also to prevent these vectors from harboring pathogens. So I'm not sure if Seth talked about it when he spoke with you guys, but a Wolbachia infected mosquito cannot transmit RNA viruses. So several important human diseases have been investigated in this case. So things like dengue or Eastern equine encephalitis, all these RNA viruses can't replicate inside of a mosquito if the mosquito has a Wolbachia infection. And because of that, people have said, oh, well, why don't we just release a whole bunch of mosquitoes infected with Wolbachia, and then they won't be able to transmit diseases. However, more troubling work from Jason Raskin's lab has shown that sometimes the Wolbachia inside of the insect actually promotes the replication of these viruses. So given that we don't know all of the viruses that are out there and all the potential zoonotic diseases that are out there, it's something to think about and something to question when we're using this organism like Wolbachia to prevent, to control vectors. And I should also say, we don't even know how this is working. We don't know how pathogen blocking works. We don't know how even reproductive manipulations work. So we don't know how easy it will be for these vectors and these pathogens to evolve resistance to Wolbachia.
So Irene, I take it then that you share my concerns about the Australian experiment. And I mean, I can make a joke and say, I think I've seen this on the Sci-Fi Channel and it doesn't end well, right? But the fact that they, and you've probably seen the wonderful animated version of this work that was all over the internet. They make little cartoons of it. And I'm thinking, and they're saying, isn't it great? And they have these figures of people smiling and all the rest. And I'm thinking, well, you don't really know what the ecological impact is going to be. No, and they don't know. They don't know what it's going to be, but I guess they kind of feel like this is such an important thing to control that, you know, it's good to have another potential weapon in our arsenal to combat these vector diseases. And there is no vaccine for dengue, and they just have 35 cases on the Big Island of Hawaii. So it's kind of transnational, isn't it? It's going to be more of a problem with global warming and climate change and all of these vector populations moving into areas they didn't normally occupy. Like we just saw the first cases of chikungunya this past season in the United States as well. So I think that obviously Wolbachia is a potential vector control mechanism, but I think we need to understand exactly what it's doing to these viruses and how these interactions with other insects and other viruses might then impact not just the prevalence of these diseases, but also the evolution and ecology of other insects. All right, so next, Talia. Did I say that correctly? Yes. All right, Talia. What scientists assume, here we are. What scientists assume is needed for life has expanded quite dramatically as researchers began to understand what life can actually exist in a range of conditions. However, we still expect the fundamentals to remain the same. Is it possible that life can exist in an entirely different form from any life on Earth? I think it is. It's possible that, and I guess it means it depends on what you mean by entirely different. So, you know, is it possible that we could have silica-based life? You know, it's very similar to carbon and probably forms similar bonds to carbon. So you could imagine an entire suite of organisms on another planet that has silica-based life. But would the fundamentals change? The physics of our universe is the same. So we would expect that things like energy and electrons are necessary for there to be life. People have also claimed that water might be a necessary requirement for life based on what we know about Earth. The second part of your question was if Riftia lacks a digestive system and instead receives nutrients from symbiotic bacteria, how do the bacteria get nutrients to sustain themselves? And how does the tube worm expel bacterial waste? We don't know. There's so much we don't know about the deep sea. And actually, this is, I've always loved symbiosis. I've always been attracted to looking at symbiotic systems because I find it fascinating to think of your ecology as another organism. So the fact that these bacteria are trapped inside of a eukaryotic cell and their environment evolves, which is crazy, right? And they have to keep up this arms race with their host every single generation to be able to persist. That said, in the deep sea, it's actually really difficult to do experiments, you might imagine. So it takes 2 hours and change to get down from the top of the ocean to the bottom in the Alvin. And then you get up at 8 o'clock in the morning on the ship and you're probably extremely seasick if you're me.
and you're kind of doped up on those anti-seasick medications, and you get inside, and the Alvin is tiny, and you're crammed in there with the other scientists and the pilot, and then it takes 2 hours to get to the bottom, and then when you get to the bottom, the Alvin has these robotic arms that are controlled by the pilot, and he takes the samples and puts them in a box, which is lovingly called the coffin, and then it takes 2 hours to come back up. And that's pretty much it. There are people like Peter Gerges at Harvard who have built these contraptions to try to maintain the pressure and maintain the temperature for these organisms, because at the bottom of the ocean, so where we were, for example, it was just off the coast of Mexico at 13 degrees north, and the temperature at the surface is, Caribbean, balmy, and then at the bottom, it's 4 degrees. And the pressure is extreme. The pressure differences are extreme. So when you passage these organisms through that distance, you are doing a lot to change them. And then the material you get at the top, it makes it hard for you to ask any real questions and do real experiments. So this is to answer someone else's question about, the switch from the deep sea to Wolbachia. This is part of the reason why I was attracted to the Wolbachia system, because we can culture it in Drosophila, we can culture it in insect cell lines. We have a huge array of different molecular tools to be able to ask questions about the symbiosis in ways that we really couldn't probe the deep sea. That said, the deep sea is still, like, metabolically the kind of the coolest interaction between intracellular bacteria and their hosts because of this kind of extreme chemosynthesis. And it's hypothesized, to answer your question more directly, it's hypothesized that the tube worms actually degrade the symbionts. That if you take a trophosome, the tissue that these symbionts live in inside of the tube worm, and that you dissect it and you fix it and then you slice it and take microscopic images of each slice, you can actually see that there's a zone at which bacteria appear to be healthy and dividing, and then there's a zone at the other end where they appear to be lysed and they lose their morphological shape. So it's hypothesized that this is kind of like a conveyor belt, that the worms are farming these microbes on the inside and that they consume them directly inside of intracellularly.
It's interesting because about 1 million years ago, Irene, when I lived in San Diego, I visited Horst. What?
I thought you were going to say something about like the Earth.
Yeah,
years ago.
Back when oxygen was first produced, exactly. No, I got a chance to visit Horst Feldbeck at SIO, and he had a Riftia that he was dissecting. And number one, what a great smell. But number two, if you look through the trophosome tissue, you see the crystals of sulfur.
Yeah.
And this takes us back to what Talia is discussing, because they take that hydrogen sulfide and they use that as a source of electrons. And what's left behind is, solid. And there's a limit to how much they can do, and they're depositing it. So this is
so specifically about the sulfur, and why isn't the sulfur toxic to Riftia, right? There's a great tradition among biologists to eat their study organisms. Maybe not so much in microbiology, but definitely true of the zoological types. And you should never eat an organism from the deep sea because sulfide is toxic to you. And the reason it's toxic to you is because it binds hemoglobin, and it binds it with really high affinity, and you basically are not going to be able to breathe afterwards. So Riftia has this amazing adaptation where its hemoglobin actually has 2 different pockets, one which binds sulfide and one which binds oxygen. And it brings them together without reacting with each other, so that the sulfide and the oxygen haven't chemically reacted. It brings them to its symbiont in that form. And the symbionts can actually use both molecular sulfide as well as sulfur. So those crystals could be used, or the form of sulfur that's in those crystals can be used by the symbiont. And it can use thiosulfate, it can use hydrogen sulfide, but it does accumulate in the tissue of Riftia. But the metazoan has kind of figured out a way to deal with the toxicity. Does that answer the question? Yeah. Okay. Braith? Hello. All right, so let's see. In light of the discovery of organisms that are chemosynthetic, do you think more organisms that have yet a different source of energy will be discovered. And do I have any guesses about what source that might be? I think that it's a neat question, and it's kind of like the Animox organisms. If you can hypothesize any potential interaction between chemicals that would liberate enough free energy for life, probably there's a microbe out there that's doing it. And so if you guys probably learned about the redox tower when you were taking microbiology, maybe? Redox tower, does it ring a bell for anybody? Am I?
Not for these freshmen.
Oh, not for these freshmen. All right, sorry. So there's this thing, this concept of a redox tower, where there are certain chemicals that are good reductants and certain chemicals that are good oxidants. So for example, oxygen is the best oxidant that we know about, and a good reductant is something that donates electrons, like a sugar. So if you combine a sugar and oxygen together, you can burn them, create energy. That's what you guys do. Bring those together and through redox actually get energy from it. So if you can hypothesize an interaction between some chemical moieties on the planet that produces enough energy for life, then there is likely a microbial group out there taking advantage of it. The question is there a symbiotic relationship that might be taking advantage of it too? And that hasn't been discovered yet, and that's entirely possible. I do not have a guess off the top of my head as to what that might be, but maybe Braith can look at the redox tower and invent some symbioses.
You know, I don't know if you saw this recently, and I mentioned this very briefly to my students and also my microbiology students, my juniors and seniors. I didn't know this. You can actually grow mealworms on plastic now, and they develop a particular gut microbiota that enables them to break it down. And I had never heard of this before.
I have never heard of that, no. Tasty.
Well, I think it just reminds us how protean, by definition, the microbial world is. They take advantage of everything.
That's true, yeah. And I mean, you select, you know, like any environment, the gut is a chemostat, and you select for whatever you provided nutrients for, you know. If you have the ability to select it, and that, you know, function actually exists in the environment. Okay, Carmen. Hi, Carmen.
Hi.
So how did I become interested in the deep sea, and what kind of bacteria grow there? So it's a good point, Carmen. Not all of them are symbiotic. All right, so there are tons of free-living organisms at deep sea hydrothermal vents, and many of them have similar metabolisms to those that the chemosynthetic symbionts have, and actually a huge number of the symbiotic organisms have a free-living state. So Riftia Pachyptila, while I was a grad student, and in Colleen Kavanaugh's lab, Colleen actually discovered this symbiosis. So she, as a grad student, discovered that these tube worms had these bacteria in their trophisome, and that was how they survived. So that's a huge discovery, right? And for a long time, because these are mouthless and gutless worms, it was assumed that the bacteria must be maternally transmitted, meaning that the moms must provide it to their children, because honestly, that's like just throwing your kids out there and saying, fend for yourselves, right? I mean, they can't do anything without their microbe. So wouldn't the best evolutionary strategy be to provide it? But as it turns out, that's not the case. The worms pick it up from the environment, and the chemosynthetic symbiont has a free living state and actually has flagella based on the genomic annotation, and so presumably can chemotax as well to different types of sources of sulfur and oxygen in the environment to be able to maintain its populations. And the same is true of a slew of other symbioses, like the mussels and the nematodes. They all have extracellular and free-living forms of these symbionts. And then the other bacteria that grow at hydrothermal vents, I should also mention the other prokaryotes, so there are also archaea that grow at hydrothermal vents, and they actually grow within the chimney of the volcano. So at extremely high temperatures and pressures, you can find these bacteria and these archaea. See, JT?
There you go, JT.
Hello. All right,
I see
so
you shaved today, JT.
Are there any other animals that use these types of endosymbioses to get nutrition and energy? Referring to the albinoconchi or albanellid symbionts. So actually, the deep sea hydrothermal vents have gotten a lot of play. So it's like a sexy environment, you know. But if you do research, it's like I went to the bottom of the ocean and there are volcanoes there and it's super awesome. And look at all these new species I found, and everybody thinks that's great. But this type of symbiosis exists in any environment that has sulfur. And a classic example is Solomia velum, which is a tiny little protobranch. So it's a bivalve about this size that you can find in mud flats off of the coast of Symbioses in Massachusetts and all along the mid-Atlantic. So it's anywhere that there is sulfur being produced and that there is a gradient of sulfur and oxygen, you have the ability to find the chemosynthetic organisms and chemosynthetic symbioses. They've also been studied in the Mediterranean as well. So you don't have to go to the bottom of the ocean to find chemosynthetic symbioses.
And we have them here, JT.
Oh, you do?
We do. We have them all through Puget Sound. And I think I mentioned to you that for almost 100 years, folks buried wood waste right around here, right half a mile from where we are right now. And you can get literally 4 millimolar hydrogen sulfide in the water column.
That's awesome.
Yeah, because when the tide comes in, all of that wood waste is like a big sponge, a giant fermenter.
That's awesome. And
so I want the NSF to take samples.
I was going to mention at this point, since JT brought this up, that at hydrothermal vents, just like my little diagram showed you, the sulfur is coming from a geothermal source. So it's the fact that you have these tectonic plates moving around, causing a lot of release of energy and heat. It heats the water reacts with the rocks, and that produces these reduced sulfur compounds. But what do you think is producing sulfur off the coast of Puget Sound? And Mark gave you a little bit of a clue. So it's biological in nature. It comes actually from sulfur reducing bacteria. So there are sulfur reducers that take oxidized forms of sulfur and make reduced forms. And so there's a sulfur cycle within those sediments off of the coast. So in some ways, it can be more limiting in terms of an energy source for those organisms that are there compared to the volcanoes at the bottom of the ocean. But it is more stable because the volcanoes, you know, they erupt and then they go away. And, you know, what happens to those ecosystems when the volcanoes are done erupting? Well, they die. Yeah, that's the sad truth. So a volcano will sometimes, like I said, multiple ways for them to die, but the volcanoes will produce the right type and the right concentration of these chemicals for a while, or they will erupt enormously and then a lava flow will kill off all of your specimens, or the volcanoes will just stop producing the right quantity and then the organisms that are there will die. All right, so Christopher is next.
Hello.
Hi, Christopher. So Christopher's questions actually made me begin the talk with my little diagram. So is that helpful, Christopher?
Yeah, that was good.
Awesome. So in terms of the mechanism of photosynthesis and chemosynthesis, let's see. So cotton.
Wave, Cotton.
Hello. Assuming life started with chemosynthetic organisms, how did photosynthetic organisms evolve? That is an interesting question. So what are, you know, some potential molecular mechanisms that you could think of for life beginning at chemosynthesis and turning into photosynthesis? So for an organism to be photosynthetic, all you need is to be able to take advantage of the energy of sunlight, right? And what's amazing is we've actually discovered, you know, in looking in the ocean and looking in other environmental habitats, we have discovered other ways of getting energy from the sun that are not so complicated as what you guys learn in basic biology. So you guys probably learned the Z scheme, and you learn about all of these different pigments, and you learn about how these electrons are harvested and used in the electron transport chain. But there's also this amazing molecule called proteorhodopsin and bacterial rhodopsins. And these are just very simple molecules that sit in the membrane and just pump protons when light hits them. So you can really imagine a way for a very basic way for photosynthesis to begin, potentially by using some of these pigments that directly translate light energy into the electrochemical gradient. And I'm sorry if that is too much jargon. I feel like, yes, now that I know that you guys don't have too much of a background in this stuff, I should probably back up. Do you guys understand electrochemical gradient? No?
Yeah.
Yes? Yes for some, no for others. So, you know, the bacterial cell is kind of like a battery, and it stores energy in terms of the differential between protons in and out. And that's part of the way that it makes ATP. And so in proteorhodopsin, we have a molecule that actually directly translates light energy into the addition of protons to that battery. So being able to store more of it. Okay, Elena. Hello. What is the most unexpected thing you have come across in your research in terms of unusual organisms, beneficial abilities of known creatures, weird or scary organisms? Interesting questions. You know, it's hard to pick like the most unexpected thing I've come across in my own research. I'll tell you a story about crabs at the bottom of the ocean, which might be funny. So when I was first starting in the lab, I didn't have my project, but we went out to sea, and I had previously, as an undergraduate, I had done a lot of work on fiddler crabs, behavioral ecology, looking at how they orient to the sun and their behavior in tide pools. You guys familiar with fiddler crabs? You have them over there? Yeah, probably. No?
Great big claw.
But because I had this affinity to crabs, I said, oh, well, I'm going to look at the symbionts that might be attached to the crabs at hydrothermal vents. And there actually are crabs that live at hydrothermal vents. They are, like many of the organisms there, sightless, so they have no eyes. And they are completely white, so they're kind of like ghost crabs. And so I had this idea that we should collect some of them so that I could then look at the microbes that might be colonizing them. And what they did in order to achieve this was to put some food in a cage and then use the alvin to take that cage to the bottom of the ocean and then pick up the cage later. And it turns out that is a really good way to attract a huge number of crabs. So they brought up this cage, and it was just full, I mean, chock full of these pretty large, I mean, they're like this big, you know, white, eyeless crabs. So that was a little overwhelming and scary as a first-year graduate student. You're like, here are your crabs. This is what you asked for. And I didn't really quite know what to do with so many samples. And so we ended up divvying them up to different researchers that were on the boat at the time. And I never did get to look at the crab-associated bacteria, but that was a scary or funny experience. Carolyn. Or Caroline. Hello.
Oh, you should ask Caroline what her term paper subject is.
Oh.
She's doing hers on Riftia.
Oh, very nice, yeah. So when I was an undergraduate, I did my term paper on Riftia. So we had, I took Amy Vollmer's class, and I don't know if you know Amy, Mark.
I do.
She does this adopt a microbe thing in her class, and so everybody picks a microorganism, and then you learn different aspects of microbiology through the eyes of that microbe. And I did mine on Riftia, and Riftia is an awesome symbiotic system. But to get to your question, do I believe that chemosynthetic bacteria may over time become like our mitochondria? Ooh. So this is another kind of big I don't know, kind of thinking question. My PhD mentor used to refer to them as sulfoplasts. So it's, you know, you can think of them as plastids to some extent, and over time that they would become like our mitos. Interestingly, as a PhD student, I worked on the genomic analysis of the first chemosynthetic symbiont that had been sequenced, that of the giant clams. So these are Clyptogena magnifica. They are dinner plate sized clams. I mean, seriously, huge. And if you cut one open, the entire thing is gill. And the gill tissue is just chock full of these sulfur crystals and of these endosymbionts. And in that system, we know that the symbionts are primarily transmitted maternally, so from mother to offspring. And in other symbiotic systems, and maybe you guys have heard of these already, you get this effect of genomic reduction in these bacterial symbionts as they are maternally transmitted every generation. And the thinking is, oh, because they are inside of this, quote, stable environment, they don't require certain things like flagella, right? They don't require things like maybe regulatory systems that are existing in other prokaryotic species. And so over time, their genomes get more and more reduced until they become something like mitochondria. So if you are familiar with the aphid-Buchnera symbiosis, in that symbiosis, Buchnera has essentially become a plastid. I mean, really what it is a plasmid that makes tryptophan, right? I mean, its genome doesn't have much in terms of content. We're talking about a few hundred kb at most. However, the chemosynthetic symbiont that I worked on has a complete genome. It can do everything. I mean, it's a chemoautotroph. So you can, what we hypothesized was the host is not eating at all, right? The host needs that symbiont to be fully capable. So in that situation, the symbionts need to maintain their functionality because otherwise they wouldn't be able to provide food for their host. So, so do I think they can become like mitochondria? Yes and no. I think that they can become really dependent on the host, but I think the host is equally dependent on them to be fully functional. So they won't exhibit the same kind of genomic reduction and evolutionary degradation over time that we see in the mitochondria.
So we're almost out of time, and I just wanted to make a quick announcement. You know, everyone has picked up handouts. We have 2 more talks to go. On Friday, we have Susan Perkins, curator of the American Museum of Natural History, and excitingly, our last speaker on Monday, you know, is Don McDonald, who is actually a real expert in the human microbiome. So I'm really looking forward to both of those. So make sure you pick those up. I also would like to take this opportunity to have everybody thank Dr. Newton for participating today. So a big round of applause. And thank you all very much, because they're anxious for their next class, which some of them are taking intro bio right now. So I would like to also take this opportunity to say how much I've enjoyed this, a chance to hear what you're saying.
No problem.
Some of the things that you're saying are things that I've said, and this is because I know Colleen, I know Amy. Amy's one of my longtime supporters, God help her. And I just want to say it's been such a pleasure to get a chance to chat with you this way, rather than extremely briefly at the ASM.
No problem.
Okay.
Thanks.
Thanks so much again. Bye now.